the Center for Drug Evaluation Research.
Thank you, Mr. Chairman, and good morning. The purpose of today's meeting is for FDA to get expert advice from the committee on a marketing application for the drug eteplirsen. And what I'd like to do is provide a framework within which to consider these data based on my 30 years of experience at FDA and really extensive experience in implementation of the legal standards for drug approval. The clinical development program for this product has features that render the data particularly difficult to interpret. It consists primarily of long-term observation of a group of 12 treated individuals. When a large treatment effect is observed, for example, significant improvement in a disease characterized by overall progression, an uncontrolled study can provide compelling data. Where overall effects are smaller, and especially if there's large inter-individual heterogeneity in the disease course, interpretation of data like this can be challenging. The sponsor and FDA have attempted interpretation by comparing the results in treated children to the disease trajectory that is recorded in a number of external cohorts. It's possible to reach different conclusions about these comparisons as is being discussed today. Eteplirsen is intended to improve outcomes in a targeted subset of DMD patients by enabling muscle cells to produce a truncated version of the protein dystrophin, which is missing or present at very low levels in patients with DMD. There is agreement that eteplirsen does achieve its primary intended pharmacodynamic effect, that is, production of a truncated messenger RNA, and this is based on PCR results from muscle biopsies. It was originally hoped that this effect would result in a substantial increase in expression of the truncated dystrophin molecule, perhaps to the average level of individuals with Becker muscular dystrophy. This has not turned out to be the case. The increase in dystrophin so far observed is a fold increase over baseline, well below the average dystrophin content in individuals with Becker muscular dystrophy. Now, it hasn't been established for any given person with Duchenne muscular dystrophy whether a small fold increase in dystrophin will provide clinical benefit, or whether there's a threshold, for example, an absolute percentage of normal, that is required to deliver a benefit. This is unknown, and of course the sponsor has just argued based on observing other mutations that perhaps small levels may be associated with benefit. It's unlikely an absolute threshold can be established given the fact that within muscular dystrophy, the phenotype, in other words, the disease expression, appears to be influenced by factors beyond dystrophin expression, so there are other factors at work. Interpretation of the dystrophin expression has been complicated by many technical difficulties. The FDA has put a huge effort into trying to render the results interpretable, along with the sponsor. To me, it is remarkable that the field of exon skipping has advanced far into clinical development generally, without well-validated methods of determining pharmacologic success, especially when assessing this biomarker requires muscle biopsies in children with compromised musculature, usually under general anesthesia. There are a lot of questions that still remain, not just about quantitating the Western blot, but also about specimen handling and intra and intermuscle variability in results, especially in later stages of disease. There are also questions, and they've been raised today, about the utility of the information supplied by immunofluorescence techniques in comparison to Western blot. And these questions are going to be quite important today. The translational science supporting these development programs is inadequate. And this state of affairs is not atypical in rare and not so rare diseases. And it significantly hinders the tasks of drug developers as well as the FDA in assessing the results of these programs. After the presentations by FDA, the sponsor, and the public, the committee will be asked a series of questions about the robustness of the data to support marketing approval, either regular approval or approval under the accelerated pathway. The determination that a drug is approvable from a clinical standpoint is a two-step process. First, a finding of substantial evidence of effectiveness, usually based on clinical outcomes, must be made, as Dr. Dunn said earlier. In the case of accelerated approval, this finding can be made based on substantial evidence using a so-called unvalidated surrogate endpoint, believed reasonably likely to predict clinical benefit. And then the second step after that is determine whether the likely benefits of the drug outweigh the foreseeable harms. And the final approval decision from a clinical basis is whether the benefits outweigh the foreseeable risks. The issue of substantial evidence for regular approval, in this case we're talking about today, turns on how compelling you find the comparisons to the external cohort data. I believe the committee has experience with this question, and so I'm not going to go into it anymore. Accelerated approval is a more nuanced issue. In the FDA Safety and Innovation Act of 2012, Congress instantiated in statute our accelerated approval regulations, and in doing so urged FDA to apply accelerated approval more broadly, particularly in rare diseases, while maintaining our standards. FDA has never articulated an evidentiary standard for determining if a surrogate endpoint is reasonably likely to predict clinical benefit. In applications of accelerated approval outside of cancer and HIV, FDA has used various types of data, including natural history data, pharmacologic, pathophysiologic, and clinical data to assist with this determination in a wide variety of settings, most of them rare disease settings. The agency has exercised considerable flexibility in applying these criteria of reasonably likely. In the case before us today, the linkage between the observed levels of dystrophin expression and potential clinical benefit will be explored. If the committee were to recommend that the clinical data represent substantial evidence, then the question of accelerated approval does not need to be taken up by you. If the committee does not make this finding, then the clinical data generated in this development program that you've heard about this morning may be used as part of the assessment of whether the surrogate of dystrophin expression at a particular level is reasonably likely to predict clinical benefit. Okay. And I'm happy to answer questions later about that statement if you wish. Finally, I would note that much of the effort in evaluating a drug development program goes into avoiding a specific mistake, that is, erroneously approving a drug that is not effective. There often is little consideration of another error, which is failing to approve a drug that actually works. In devastating diseases, the consequences of this mistake can be extreme, but most of these consequences are borne by patients who traditionally have little say in how the standards are implemented. The accelerated approval program includes a requirement for confirmatory studies for efficacy. So, as you've heard from the sponsor, you have to do further studies to explore and confirm effectiveness. An inherent presumption in this program of accelerated approval, which is written in the preamble to our regulation about it, is that more uncertainty is going to be tolerated initially, and that in fact sometimes we will collectively get it wrong. Otherwise, accelerated approval would really have no different standards than regular approval. I hope these remarks have been helpful, and I look forward to hearing the deliberations. Thank you.